So good morning. Welcome to the 49th edition of the National Meeting for Society of Toxicology. I'm Kyle Collegier. I'll be chairing the session today with Chris Parker from Cellular Dynamics. The workshop session is entitled Toxicology in the 21st Century, the Present and Future of Stem Cells in Drug Discovery and Drug Development. Because of the rigorous schedule that we have, I won't have a real long introduction, and Chris will actually give a lot of the background on value and the interest around stem cells as they relate to drug development. And we have a really exciting set of speakers. We're really looking forward to a robust discussion. And with that, I introduce the first speaker, Chris Parker. Chris's title of his talk is Human Inducible Pluripotent Stem Cell Derived in Vitro Models, the Path to a Better Understanding of Individual Biology and Their Utility in Drug Discovery and Development.
Well, thank you for that run-on sentence title that I came up with. Welcome to the session. I hope we find all the different talks elucidating and interesting. I think we're at a very exciting time in biological discovery, and there's no question that the potential of stem cell and induced pluripotent stem cell models and therapies are at the forefront of where biological research is moving today. I think there'll be a number of discussions by the other speakers about exactly where the potential lies on some of the therapeutic applications and where the field is going in general. What I hope to do is give you a little bit more of a practical presentation on the potential of stem cell-based tools in drug discovery and development as model systems from an actual tool developer. We're a company that is trying to leverage stem cells and produce cellular models for research, and I wanted to go through a little bit about what are the next steps in terms of being able to deliver on that. The agenda for my presentation is to give you a brief stem cell history. We can do that because the field is relatively young, and although there's been a lot of recent activity, the original field was initiated in 1981 by Sir Martin Evans. Then I go into a little bit of who we are as a company, CDI, just briefly, and then talk about the tool requirements and really what's necessary to leverage stem cells as it relates to cellular tools, and why we've elected to use IPS cells, induced pluripotent stem cells, as a starting material for development of those tools. And then some more practical constraints on coping with the challenges of stem cell tool development. Hopefully this will be interesting from a standpoint of not only understanding the process of stem cell differentiation, but also some of the things that we've done to be able to make sure that we can produce reliable tools. And then what can be produced and what we're making today. And then what's next? What are the next steps in this field? And I think some of the other speakers will actually elucidate that even further. As I mentioned, in terms of the brief history, the stem cell field was really originated by Sir Martin Evans in 1981 when he was able to culture mouse embryonic stem cells in a pseudo-uteral environment, and from there the field has really taken off quite dramatically, and a lot of mouse stem cell work and other derivations of stem cells was happening during this time. And then in 1995, Dr. Jamie Thompson at the University of Wisconsin was able to establish cell lines for a variety of different primate species to make sure that species diversity could be maintained over time. And of course then shortly thereafter the cloning of Dolly the sheep, utilizing stem cells to provide the material to clone that sheep. And then a variety of different publications in terms of now starting to leverage the stem cells as a starting material into a variety of different tissue types, including hematopoietic cells and a variety of others, including cardiomyocytes back in about 2000. Our company was started in 2004 based on the work that Dr. Thompson had done and really the practical nature of how stem cells could help medicine today. There is obviously long-term perspective from a therapeutics application, but to really provide practical methods of studying biology today was really what our focus was. And then simultaneously in 2007, Dr. Junying Yu in the laboratory of Jamie Thompson at the University of Wisconsin, as well as Shinya Yamanaka at Kyoto University, were both able to simultaneously publish papers on the creation or reprogramming of induced pluripotent stem cells. They use similar but different factors to reprogram somatic fibroblasts back to a pluripotent state. And then In 2009, in April, Junying Yu, and again in Dr. Thompson's lab, demonstrated the first vector-free or residue-free methodology of inducing pluripotent stem cells. The first in 2007 used a retroviral vector that integrated into the genome of those cells. And then in 2009, we were able to produce iPS cells from those from somatic cells without the introduction of a vector. She utilizes an episomal approach. And then in 2009, a full mouse was created from stem cells in its entirety. It's the first indication that an entire organism could be created by the use of stem cells. And now I think really is you're going to see an even more acceleration of research in this field, not only on the reprogramming side, but also in the differentiation process of taking those cells and moving them into terminal cell types. What CDI is a life science company focused on the development of iPS cell-derived models for research. We're headquartered in Madison, Wisconsin. We have 73 people currently in the company. We occupy about a very cramped 25,000 square feet. We're literally busting at the seams. And we have a GLP production facility, R&D and manufacturing labs, and clean room for automated cell culture to produce large quantities of stem cells. And our emphasis is really on product development and manufacturing of terminal cell types using iPS cells as a starting material. And we've really established what we believe is a manufacturing platform for large-scale production, both of the parallel derivation of footprint-free iPS lines, as well as the manufacture of differentiated human tissue cells, including cardiomyocytes, hepatocytes, neuronal cells, and endothelial cells. And I'll go into a little bit more detail as we enter into the presentation. The company was really founded by Dr. Jamie Thompson and a number of the collaborators that he had that really understood the ability to use stem cells as a starting material to understand organotypic biology and develop tissue models that could represent better models than were existing currently. Dr. Craig January, for instance, was the person that originated the human embryonic kidney herd line that many of you use in your herd screening activities. Dr. Tim Camp also worked on was a noted cardiologist at the University of Wisconsin, also was one of the first to demonstrate the differentiation of embryonic stem cells to cardiomyocytes. And Dr. Igor Slurpman is a pathologist that has an expertise in hematopoietic cell differentiation. And really this was about how you could take a practical approach and really deliver tools that could impact drug discovery and development today, understanding that would affect human medicine quickly as opposed to 10 to 12 years. Now, when you think about a cell-based tool that can be predictive of human biology, there's a couple things that are obvious that I think need to be stated, and I actually presented this over 2 years ago at a CERN meeting, and they haven't changed. I think really the nature of any tool that's developed for research is that it is predictive of an in vivo response, that it correlates with existing in vitro and in vivo models. If you have a model that doesn't correlate, you question which one is accurate, that these cultures can be reproducible and homogeneous such that you can do longitudinal studies and create a compendium of knowledge to base future predictions on, that they are validated in chemical space. And I think this is one of the things that we think is one of the most important as it relates to tool development, that they respond as the cell in vivo model would from a physiological perspective. When you induce or perturb the cell with for instance, cardiotoxic compound, that the cells respond appropriately, and that can be validated over a variety of different compounds, and also that can be correlated via dose and time. And then, most importantly, that these cells can be generated in sufficient quantities such that they can be widely applied in a variety of different applications. I think, coming from the tools industry, the measuring devices currently available have now outstripped the cell models that they're actually being used to test. And providing a better model will make those tools even more powerful, including genomics and proteomics and cell-based assays and high-content imaging, because we're looking at a real human biological system. And then I think in the long term that they potentially have the acceptability for regulatory agencies, that the data can be trusted and be reproducible from a drug filing perspective. And when we think about in vitro models, most of the models that have been utilized are either transformed cell lines that are basically immortalized cells or cancerous cells that have been derived from a given organotypic tissue, but they are generally genetically unstable. They obviously are in logarithmic growth, so they don't really represent true biology. Their quantities are unlimited, but they change and they have a tendency to drift in culture if they're maintained for long periods of time, and their gene targeting is very limited. Primary cells have a limitation by batch-to-batch variability, a quantity issue, and really whether those primary cells are representing the biology of the tissue that they've been perfused from. But pluripotent stem cells provide us a diploid environment that has stable growth. Really, the quantities can be unlimited in terms of starting materials for differentiated cells. Their cellular variability, at least to date, is relatively stable, and they have the ability to do gene targeting, and I'll show you that as we move forward in the process that we utilize at CDI. And so, one of the things I wanted to make sure people were understanding of is this difference between embryonic stem cells and induced pluripotent stem cells. And I think this is one of the most active fields today in the stem cell biology field, with finding different ways to reprogram somatic cells into pluripotent stem cells. The methods originally were the introduction of a transient set of genes that would induce reprogramming of those cells back to a pluripotent state in the same way that the embryonic stem cells came to the culture of a pluripotent state. Those cells then can be utilized as starting material to differentiate into a variety of different cell types, including any really in the body. And I think there's been a lot of activity to use different methods to induce these pluripotent stem cells. Our focus at the company is really to find ones that are residue-free, that are safe, that can be put into a highly parallel environment, and that are robust, such that when the reprogramming process is done, that we have a characterizable induced pluripotent stem cell population. We've actually settled currently on an episomal approach that was derived by Junying Yu, who has recently joined the company as a scientist, that really introduces a plasmid vector that doesn't integrate into the genome and does its machinery work, provides the gene materials that causes the reprogramming, and then puts them into a pluripotent state. But this field will continue to grow. You're seeing things from small molecules to proteins to other types of vectors, and we know that this research will continue. And when these induced pluripotent stem cells are created, the idea is to compare them to the base case of embryonic stem cells, and we've done a lot of work to make sure that, for instance, that these cells, as embryonic stem cells, have a normal karyotype, that they cluster by gene expression profiling with the appropriate embryonic stem cell or pluripotent stem cell gene signatures. And we've demonstrated that with the variety of different IPS lines that we've created at our company. We've now currently been, I think, well over 50 different IPS lines from a variety of different tissues have been generated, and all those still fall into these categories. They have the typical embryonic stem cell or pluripotent stem cell morphology with a large nuclear to cytosolic ratio. And that when we look at cell surface markers comparing embryonic stem cells, that they have similar patterns of pluripotent markers on the cell surface that can be measured appropriately. And really, the reason why we chose IPS cells to focus on is, I think, relatively obvious from the standpoint of being able to represent individual biology. But I think the applications really run the gamut from basic tool research, basic cell biology research, all the way through personalized medicine applications. Some of the areas that we're focusing on are the concept of being able to represent individual biology, to be able to look at how an individual responds mechanistically when we create their derived cell types. And I think one of the really important areas is the idea of creating panels such that we can better represent in vitro or clinical trial populations. We actually call it an in vitro clinical trial that we can represent, for instance, 8 different ethnicities with half male and half female, and then derive IPS cells from those, and then a variety of different cell types from those individuals to really understand how those particular groups will respond to a molecule as opposed to a generic model, as well as looking at genetic predisposition. We currently have a collaboration with the university to generate IPS cells for individuals with genetic long QT syndrome and be able to provide cardiomyocytes based on those. It also affords the opportunity for different disease models. This is an area of active research as well, because it's unclear as to exactly what diseases will manifest in the dish when an IPS cell is generated from those individuals. But we also have the ability to engineer these cells to overexpress or underexpress a particular disease-based target that can be used for a research tool. And of course, in the long term, I think you have the ability of personalized medicine and applications in terms of being able to generate cell types to test drugs on before an individual takes them. I think that's still a ways away. And obviously, the potential of autologous cell therapy where there'd be no impact on rejection by a given individual. And really the idea is that we can represent an individual's biology by their IPS cells and then represent any cell type in the body, some 200 cell types from that same individual, so that we can really start to understand system biology as well as individual biology. So when we talk about the practical constraints of generating these tools, there's some components that really become critical, one of which is that you have to have a consistent, high-quality, large-scale stem cell culture. It seems trivial, but it really isn't. And in fact, making sure that you have consistency and that you can prevent the cells from differentiating as they would like to do. We're basically training the cells to maintain their pluripotent state and preventing them to do what they want to do, which is to differentiate into all the cell types in the body. It's important to make sure that that process is very well controlled and that we eliminate variable components in terms of materials that would be different from batch to batch, and that we have limited human intervention so that we can scale it to sufficient quantities. And we've really made a lot of strides at CDI to be able to do that. I think the next thing that a lot of people are working on is the optimization of differentiation protocols. Because these cells really will differentiate into any cell type in the body, they do it at very low yields. And the ability to drive that beyond a basically random differentiation process is really critical to get sufficient quantities of these cells to be able to be useful. And that is really to focus on increased yield. Most of the written protocols in the literature demonstrate maybe anywhere in the single-digit percentage yields from a given IPS cell. And then I think the important thing, again, is to make sure that when you do these differentiation protocols that you have batch-to-batch consistency and that the cell populations are consistent over time. And we've made some great strides in industrializing this process and getting these yields up into the double-digit percentages such that we can actually generate large quantities of these cells. I think the next thing is purity. Again, because these cells differentiate into all cell types in the body, they also produce other cell types in these cultures. And this becomes important if the purity is low, such that you're really understanding that the cell type of interest is really driving the biological response and not some other contaminating cell population. And via the manufacturing method that we utilize, we can actually maintain purities of our cells up to 99%. And also that these cells can then be maintained in long-term culture, such that these other contaminating dividing cells, because most of the cell types that are differentiated are terminally differentiated and by definition don't divide, the other cell types that are present in these cultures have the ability to divide and it will eventually overrun the cultures and take over. And it's important to make sure that when you have that cell population that it maintains itself for a long period of time. Our current protocols allow for our cells to maintain their purity for over 2 weeks. And then I think the obviously is the industrial scale. It's easy to produce 1 million or 2 million cardiomyocytes using literature protocols, but when you're trying to generate billions, it's a totally different scale. And the numbers and types of expertise that are required change from just cell biology to employing all sorts of other functions, including process development, automation, process sciences, and the like. And I think the next steps are really to focus on the parallel residue-free IPS reprogram process. We're not there yet. I mean, this is one of our major focuses in the company to be able to generate large numbers of IPS cells simultaneously, but this is our focus. We have an approach now that's robust and that can be used to generate IPS cells, and we're going to continue to try to leverage that and scale that such that we can generate, 96 IPS lines from 96 different individuals simultaneously. And I'm confident that we're going to get there. And then I guess the next thing is to really understand how to characterize those individual clones of IPS to make sure that they are similar in their pluripotent state. And I think there's going to be a lot of research in this area to really understand the differences between IPS lines as they're created, not only from different cell types. We currently generate IPS cells from fibroblasts, keratinocytes, as well as circulating peripheral blood. And to be able to understand exactly how those are similar or different is going to be important as they go into the differentiation process. And as I mentioned, I think the continued improvement in IPS characterization is going to continue over time and will be a major focus of many researchers as these numbers of lines increase. The way we do this is really by generating a pipeline, a manufacturing, industrial scale pipeline that's managing stem cell biology, and you can see here that we're broken down into a variety of different groups. Everything from research in terms of reprogramming, that group is led by Junying Yu, who came out of Dr. Thompson's lab, and we have 14 scientists working on reprogramming and understanding how we can use different tissues and make that process more efficient. And then really the optimization and genetic engineering of starting material to produce a given differentiated cell type is done by a variety of groups, and we then isolate those groups based on cell type And then we have a process sciences group that is really about scaling the manufacturing in a lot of the same ways that bioprocessing is done in the pharmaceutical industry, but with an eye that stem cells are different than transformed cell lines in terms of their propagation and their proliferative capacities. And then production, where once these processes are produced, that we have a production group that can produce large numbers of them and be able to cryopreserve them and maintain them for use. And I put this slide up primarily because we've put a quality system in place, and we've noticed that by managing the materials, that by managing the training, by managing that there's consistency among people and equipment, that this has really made a dramatic impact on our yields as well as the quality of the cell types that we're producing. Some of the things that are interesting about this is that in some of the supply quality management issues, we're actually purchasing quantities of materials that are used in the differentiation process that manufacturers have never made before. So making sure that they are able to consistently provide that material for us to manufacture is really critical to be able to create consistent cell populations. Our process is really driven by a genetic engineering approach that is proprietary to CDI. We use a homologous recombination in the IPS line, the initial starting material, that has a cell-specific promoter that's attached to a fluorescent protein tag and an antibiotic-resistant gene. This then allows us to very quickly iterate the process. For those of you that don't know, the hallmark indication for cardiomyocyte differentiation is around day 14 when cells begin to beat. That's very problematic when you're trying to optimize the process and increase yields, to wait for that period of time before you know whether you've changed or improved the yield. By having a fluorescent protein incorporated into these cells, we can actually see as soon as that percentage of quantitative fluorescent protein comes up, we know we've impacted that yield and dramatically. And then what it also provides us is a way to purify that cell population by applying antibiotics and getting to a cell purity of about 99%. But really the next step is to make billions of stem cells, and hopefully I mentioned before that's not a trivial milestone, that we're actually producing over 1 billion IPS cells per day that go into as starting material for these processes. And as I mentioned, when your yields are in the 15 to 20 to 30% range, you need a lot of starting material to make sure that you can provide sufficient quantities. And then they go into the differentiation process, and then finally the purification process. And this is an example of our first product, which is a cardiomyocyte product that is a pan-cardiac specific population of atrial, nodal, and ventricular cells. And you can see here on the upper left, these are grown as aggregates. Each one of these aggregates is about 1,000 to 2,000 cardiomyocytes. You can see in the pre-purification that we have large colonies, but in the right, you see that not all of them are lighting up red, but a preponderance are. In this particular instance, we were at about 13.5 to 14% yield. And then when we apply the antibiotic, you can see, again, the populations are similar, almost identical to the populations here, so that we get the purity that is required to cryopreserve at the end stage. And this process is turning out to be amenable to a variety of different cell types that we can put into this engine that we've developed. And really, so if you were to use these cells, they come as cryopreserved single-cell suspensions at about 99% purity. The vials contain sufficient quantities to populate a device of interest, whether it be for high-content imaging or cell-based assays. They're shipped on dry ice, and they have long-term storage in liquid nitrogen, and they're ready to thaw and use, and we actually provide a plating media and a maintenance media that will maintain the purity out over the 2-week period that we recommend. So, this is, to our knowledge, the first product that's been generated by IPS cells, and it's a field that's really only 2 years old in terms of the first reprogramming. And these cells, have the functionality of normal human cardiomyocytes. They have action potentials that are replicative of normal heart biology in terms of atrial-like, nodal-like, and ventricular-like. They have the appropriate ion channels and can be impacted by appropriate drugs that are known to block those channels in terms of nifedipine and in terms of calcium channel, and also potassium channels, looking at E4031, and that we can see these EADs that are formed. In addition, they respond in a dose-dependent manner to known cardiotoxic molecules, and that we can identify IC50s that are relatively physiologically accurate. As I mentioned before, we could say that we have cardiomyocytes and they have all the cell surface markers that make them, look like a duck, but until they start quacking like a duck and responding to known pharmaceutical agents, they really don't have the same utility as a good tool would have. And some recent data that has been generated to show the stability of these cells by our counterparts at ROS show that we're seeing from global gene expression profiles that the stem cell genes are being down-regulated during the differentiation process and remaining off for the longer period of time. These cultures are actually looking at the differentiation process from day 0, which is at the IPS state, all the way out to 120 days post-differentiation. You can see that there's a number of genes that are being actively turned on during the differentiation process that can be mined for optimization. And then the most important thing is that when these cells start to go through the differentiation process, right about day 14, which is when they start beating, that those hallmark genes of cardiomyocytes stay on for the entire 120-day period, demonstrating the stability of these cells in culture. We actually have had cardiomyocytes in culture that are beating from these cells for going on 6 to 9 months that continue to just beat by just changing the media. So what's next? Now that we've got this, kind of pipeline and we got a handle on how to differentiate these cells, I think we're going to go down the path of some of the cell types that are of continued interest to folks in the pharmaceutical world. Obviously, hepatocytes. This is from recent feasibility data that we've generated. You can see here the purity ranges are low, but this isn't really daunting for us because our purity ranges for cardiomyocytes were much lower than this, so this is very encouraging that by employing our genetic engineering methodology that we're going to be able to increase these yields. But so far they have this similar hepatocyte-like morphology, and they are expressing albumin with the appropriate markers. We've also demonstrated pan-neuronal populations, feasibility data where we've got astrocyte formations. We've been able to identify GABAergic, dopaminergic, and glutamatergic neurons in these cell populations, and we're going to continue to move those into product development as well. We've also demonstrated some utility in endothelial cells that can be used potentially for angiogenesis research, as well as the understanding of the blood-brain barrier, as well as their activity in a variety of different autoimmune diseases and inflammation responses. And they have the typical angiogenesis formation, tubular formation, and stain appropriately for von Willebrand factor and acetylated LDL, although our negative control was not the best choice in those particular feasibility data. But all three of these have achieved our bar for feasibility, and we're moving forward to try to manufacture these in numbers. So, conclusions. Hopefully what I've shown is that IPS cells can be utilized to generate large numbers of highly pure cell populations, and that these could potentially be very valuable models in the drug discovery and pharmacology realm. I think that there's one thing for certain, that IPS cell technology will continue to evolve. I think you're going to see the numbers of lines overall increase. I know the NIH has activities to generate over 1,000 lines, and Cornell and Harvard and a variety of others are moving forward with generating large numbers, so there'll be a plethora of starting material to leverage. I think also we'll be looking at additional, again, we've already done it from skin, hair follicles, and blood, and I think people will be looking at other tissues to reprogram from as well. And then I think for us, in terms of where this is going, what are the next steps? And I think the first thing that questions occur after we say we can derive and deliver a pan-cardiac population is, well, can you make ventricular-specific cells? Can you make atrial-specific cells? And that's on the horizon for us as well, to go down those paths of generating ventricular or atrial or dopaminergic neurons or serotonergic neurons by leveraging the differentiation process. I think one of the other things, because of the nature of these cells being embryological or neonatal, that really understanding the maturation process and whether there's ways to accelerate that process are going to be areas of active research by us and others, I imagine, because we really can't get into the wine industry here in terms of waiting 35 to 40 years before these cells start to respond or replicate an adult population. We know they respond appropriately now, but understanding that phenomena would be really valuable in the long run. I think also, as I mentioned earlier, understanding how polygenic diseases can manifest in the dish is going to be an area of ongoing research that we're going to try to understand. And then organotypic biology. These cells are two-dimensional and are grown in flat cultures, and they're in a cardiac population where there's atrial and nodal and ventricular in a variety of different spatial areas on the dish. And what can happen if we can admixture those and create a ventricular specific population and a atrial specific population such that they can have a more organotypic environment. And there's a lot of 3D culture systems that are out there that are trying to be developed to maybe leverage this. And I think the multicellular systems aspect, because now we can represent individual biology, we can start to think about systems biology. Wouldn't it be great if we could have, for instance, a hepatocyte culture and a cardiomyocyte culture that could be put in proximity where a drug could be put through the hepatocytes and allowed to be metabolized accordingly and allow that molecule to then, in its metabolized form, to have an impact on a cardiomyocyte population, for instance. All things that can leverage other technologies to put those cell populations in proximity, but the key being that those populations are at the appropriate purity and appropriate number to be able to actually do studies. And so with that, I'll finish my talk and just say that I think the potential is, and the horizon is very good for this technology. We're excited, and I just really challenge all of you to put these types of models through the paces and make sure that they're representing better biology in your arenas. Thank you. I'm happy to take any questions if you care.
Jim Trosco, Michigan State University. Can I ask you a philosophical question? I am on touch of IPS research today, but we were the first lab in the world to isolate human adult stem cells. Mm-hmm. So my philosophical question is, why go through all of that complex process of isolating iPS cells? Why can I isolate from every in the tissue the adult stem cell?
Well, I think because they have to be isolated, I mean, my comment to that philosophically is that we want to have a starting material that we can differentiate into any cell type in the body. All those require the harvest of those adult stem cells from those individual tissues, so they have to be able to be derived from that individual, and they're in relatively low numbers and have limited expansion capabilities. iPS cells have the ability to expand almost indefinitely to produce billions of cells that can be used as starting materials. And we only need a small amount of blood from an individual to derive and reprogram those cells.
I understand that assumption, but one of the nice things about adult stem cells, you don't have the problem of inducing teratomas. And you also can expand these individual adult stem cells indefinitely if you know how to grow them.
Right, but those activities will have to be done on a tissue-specific or tissue-by-tissue basis. Other questions?
Ron Heinz, College of Wisconsin. Do you think that this technology will ever be able to reach the point where you can have the genetic diversity, for example, even in the genetic diversity in the pharmacokinetic processes, that you can recapitulate and actually do the virtual clinical trial or in vitro clinical trial that you're talking about?
I think the easy answer is it's going to be difficult. It's because we can only go on a cell-by-cell basis. We'll be able to represent that individual biology, but all the other components that are involved in vivo response, the tissue-to-tissue interaction, that's going to be years away from being able to be accomplished. I think the first step is really, can we understand how ethnic or genetic diversity applies to one cell type? But I think this is not going to eliminate in vivo models, nor will it eliminate clinical trials. It will hopefully make the process to get molecules into that clinical development pipeline more efficient, and that we'll have a better understanding going in about safety issues and efficacy issues.
Just a quick question. Can human adult cells, stem cells, be used to generate iPS cells to avoid that issue of tissue by tissue, as you were just explaining? And my second question is, how you culture iPS cells? Are they done on feeder cells, or is it standard normal cell culture?
We're one of the things that we've gotten away from is using mouse or MEFs, because that's one of the variable components that is part of kind of problems of being able to get large-scale culture. I think for the most part, the reason that the reprogramming has been used in the cells to date has been because they have a proliferative capacity. That machinery is required for the reprogramming process. I think I'm probably not the best person to ask that question. I think maybe Dr. Thompson can answer that question during his talk in terms of why we choose these somatic cell types as opposed to adult